Okay, good evening, everyone. In the last class, we were trying to discuss about some of the things that are useful for studying epigenetics. And in that, we are trying to learn about the techniques involved in imprinting, studying imprinting. So we are going to use the Southern blot technique, and we are also going to use restriction enzymes. And in that, I think we already discussed that restriction enzymes can be methylation, based on methylation of the target site. They can be insensitive, or we'll start with sensitive, meaning that if the restriction site is methylated, then some of the restriction enzymes fail to cleave their respective restriction sites. Some of them are insensitive, meaning that they would cleave irrespective of whether the DNA is methylated or with that particular site, restriction site is cleaved, methylated or not. And there are others that are methylation dependent, which would mean that they would cleave if only the DNA is methylated, otherwise not. And briefly, restriction endonucleases are the ones that break the phosphate backbones. That means they are endonucleases. And second thing is they are sequence specific. They do not cleave randomly anywhere. They would cleave only at specific sequences. Those are referred to as the restriction sites. For EcoR1, this is the sequence, GAA TTC. So just quickly to go about with these sensitive, insensitive, and dependent. I will just give two sets. You can take off, this is methylated, and this is unmethylated. Same is the case with all. Whether it will cleave or not is what we are going to see. We are just going to describe it this way. Probably it'll better the understanding. Sensitive one will fail to cleave this, will cleave the unmethylated one. Insensitive one will cleave both, irrespective of the methylation. And dependent one will cleave only the methylated target site, but not the unmethylated one. Okay. And we discussed about Southern blotting. Is that right? I hope you remember about the Southern blotting. The objective of Southern blotting is to find out if a particular sequence is present or not. And it works on the principle of hybridization, or based on the hybridization between the target sequence and that of the probe. A probe is a sequence that we are adding with a specific sequence that we know. Say, for example, probe for gene X. That means it has the sequence which is identical or complementary to that of gene X. It's usually radio labeled so that it will help in detection at this stage, at the last stage. Digestion with restriction endonucleases, electrophoresis, then it's followed by transfer, or that is the blotting. And then hybridization with the probe. We will add a probe that is radio labeled, and then eventually we will check the autoradiograph. If it gives a signal, say for example, A and B, and A did not give any signal and B has shown a band, that means the probe we added for, assume it is for gene X, then we can say that B has the sequence gene X, but A does not have. Okay? That's how, that is the basis of molecular, the Southern blot in simple terms. It look like this, and upon development of the film, it will look, the bands will show up like this. I think we also discussed this one yesterday in the last class. Is that right? Somebody, please.
Yes, sir. Yes, sir.
Yes? Okay. So we are deployed. So we may have also multiple copies of a gene, like alleles, allele 1 or allele 2, and we may be homozygous or heterozygous. So based on that, it can give rise to the banding pattern will be dependent on the heterozygosity or homozygosity, first point. Second thing is about the sizes of the fragments that is a result of restriction endonucleases. I'll just put as RE. And the third is also about the coverage of the probe. How much probe is being used here? What is the length of the probe? Right? Did we do an exercise on this, or we will just quickly do one thing? Okay, or we'll just move ahead and then we will check. Okay. Okay, I will do something else. So I want a quick answer for this and try to see if you can help me out. Help me out, figure the answer for it. So I'm going to make a new probe for the same experimental setup, but I'm going to have a probe is going to be a little bit different. Only the colored region is the probe. Say, for example, from here to here. This is the only colored region. That is the only probe. Probe fragment is, the size of the probe is there. That means it is complementary to that part and nothing else. If I did a PCR, the Southern blot analysis, and if we have samples A and B and C, and the difference is A is homozygous for allele A and B is homozygous for allele B, allele 2, and C is homozygous for 1 and 2. So what will happen? What kind of bands will form in this stage? Anybody? Please try to answer. A, what bands will it give? A is homozygous for allele 1. Okay. Anybody? Can you try? That means the person has 2 copies of allele 1. So allele 1 will be cut here, here, and here. And the second allele is also 1, right? So it will also be cut here and here. So you'll get a 3 kb fragment and 2 kb fragment. But what will be detected in the after hybridization and development of the film is dependent on where the probe binds. As per what is given here, the probe is going to bind only to the fragment 3 kb fragment, right? So which ones will show up in the band? How many bands will we see?
1.
What will be the size of it?
3 kb. 3.
Yes, you're right. So we'll find a 3 kb fragment. What will you find for the sample B?
1 strand at 5 kb.
Yes, 1 band. Okay, we'll say it as band. Yes. So we'll only find 1 band, and that will be of 5 kb in length. And what is for C, which is heterozygous, allele A and allele B?
2 bands.
Yes.
3 and 5.
That's right. So yes, thank you. And it will be something like this, and the probe will bind somewhere here. So this is 3 kb, 2 kb, and 5. Only 3 and 5 are going to be bound by or hybridized to the probe. So we'll find like this. Then too we can figure out if it is heterozygous or homozygous. Okay. So we are going to use all these things together to find out the methylation status, methylation or the epigenetic footprint as a base of methylation. So DNA can be depending on the situation, it could be methylated, something like this. It could be completely demethylated, and the other one is state of being hemimethylated. This is based on strands if you're talking about. Okay, top strand, bottom strand. If we are talking in terms of biparental alleles and so on, okay, then it is possible that both can be methylated, or both can be demethylated, or one can be methylated and the one allele could be methylated, whereas the other allele can be unmethylated. Most of these fall into the category of imprinting. I also want you to draw your attention to something here. Sorry, to go back. So we are going to choose some restriction endonucleases, and we are going to base it based on the sensitivity, insensitive, dependent, and so on. And see, if you look at GAA TTC, which is for EcoR1, there are no GCs. GC, GC, CG, like that. If you have more of them, then this is something like the sequences that we would find in CPG islands. And that is where the cytosine is usually methylated. So EcoR1 as such is not very useful directly, right? Because it's filled a lot of with AATTs, and G and C are separated. Of course, it can still be useful based on what are following or preceding it. So the choice of restriction endonucleases for this analysis, for epigenetic analysis, is that they would choose something that would have a lot of GCs like this. The mark, this symbol here indicates the point of cleavage. Okay, not one, it is a 8 base cutter. That means it is recognizing total of 8 bases. Okay, normally normal ones that are usually the ones used are 6 base cutters. And it is methylation sensitive. What does that say? That means if there is the not one site and it is not methylated, then cleavage will occur. But if that not one site, if the cytosines in that state, in the site are methylated, then it cannot cleave. Okay. So based on that, we can study the DNA methylation patterns, and it will we are now in the focus point of it is the choice of restriction endonucleases and cleavage, or the cutting of the DNA by the restriction digestion by the restriction endonucleases. So the objective of study of the actual paper is to study the methylation state. There are 2 points. One is the methylation state, and then to also study the chromatin structure by chromatin sensitivity, which we will discuss in the next class maybe, or in this class itself. And they are mostly involved, they were studying about 2 genes, H19 and U2AF1RS1. So they can work with pluripotent germinal cells, which they can derive from the embryos. They can also isolate spermatogonia. That is also possible from, you can isolate from animals. I mean, these are conducted on mouse or mice, and mature sperm cells too. It's not easy to get from eggs. Sperms usually are produced in a large quantity, so, and its ease of extraction is also, it is actually easy to extract them, whereas egg, there are not as many, and it would be difficult to analyze it as well. So the study would employ the model organism is mice or mouse, and mice, and then they would take different samples and then see the methylation status. So you have to remember that they are biparental, diploid, and one would be, if it is methylated state, it would be methylated, one copy, and the other copy would be unmethylated. But this state will depend on the status of the, or the growth state. Say somatic cells are biparental, in that you can expect one methylated, one unmethylated. During primordial germ cells, I hope you remember that the imprints are erased. In that case, we should expect both unmethylated. And during the gamete formation, what we know is imprints are established based on the whether it is a male or female. The gametes would either be methylated or unmethylated. Okay, I hope you can categorize, you can remember this like this: methylated or unmethylated. In the first one allele would be methylated, one allele will be unmethylated. In primordial germ cell stage, both will be unmethylated. In gamete stage, based on the sex of the parent, either it will be methylated or it will be unmethylated. Okay. Yes. So this is what we are talking about, the standard model. That means people until then, also even now, the general model is that primordial germ cells, during that stage, there is erasure. That means there are no imprints in this stage. And when the sperm is formed, imprints are established. As I said, they were discussing or working on two genes, U2AF1RS1 and H19. These are intronless genes, gene present on chromosome 11. You don't have to mug up all these things or memorize, but it is just for the knowledge sake, it is a part of a SNRP, small nuclear ribonucleoproteins that are involved in splicing. So typically what is known is the maternal allele is imprinted, that means suppressed, and the paternal allele, it is expressed. H19 is a gene that is present on chromosome number 7. It also encodes an RNA. And so, okay, U2AF1RS1 encodes a protein. Okay, H19 is a RNA gene, RNA encoding gene. And it is involved in embryo development and so on. The important one I wanted to talk is the paternal allele is imprinted and the maternal allele is expressed. Okay, they are opposite, right? Remember that imprinting part. So it's the same thing. And if you are looking at a biparental organism, say they are on different chromosomes, but for the understanding, I will put it as this: AF1RS1 gene and H19. And if it is paternal allele, H19 would have been repressed and U2F1RS1 would have been expressed. If it were the maternal allele, then U2AF1RS1 is repressed and H19 is expressed. Okay, so what we would generally assume is that if it is in the sperm, U2AF1RS1 should be in a state of methylation, because erasure would have occurred, and during spermatogenesis the imprint would have been established, because of which, and U2AF1RS1 is supposed to be unmethylated in the paternal allele. So in that case, U2AF1RS1 should be in a state of complete demethylation. Whereas H19, it is paternally imprinted. Therefore, in the sperm, as I said, these people were working only on sperm because it's easy, and the egg would have been very difficult. So H19 should have been methylated in the sperm. Okay. So that
So
would have been, yes, please.
I don't understand how, because methylation and
Yes.
demethylation are sex related. So without
Yes.
con- okay, you're saying got it.
Okay. I'll try again. Okay. So this is an organism, and assume that the germline is forming primordial germ cells. What would have occurred? We will call this as methylation here, and here this is methylated, U2F1RS1 in the maternal allele. So all of them should have been unmethylated. Okay, U2AF1 and H19, they are unmethylated in state, right? And during the formation of gametes or sperm here, what should happen? In that case, U2AF1 RS1 should remain as it is, unmethylated state, whereas H19 should have been methylated. Because this is sperm and this is where erasure would have occurred. So when the other one, what will happen to the other strand, other allele? Will also have the same thing. The imprint, this organism individual is a male. Okay, sorry, this is a male. So the sex of the parent is going to determine what is going to happen here. In this case, H19 will be methylated in both or all the sperms produced. All the sperms were haploid. In all of them, you will find U2AF1 RS1 unmethylated state and H19 in unmethylated state. At least in theory, that's what it is. Is this understood a bit?
Yes, I forgot the fact that they were males.
Yes, that's all right. I might have not mentioned that. Okay. So there are several other setups that we will need to understand. There are NOT1, HPA2, MSP1, BST11, SAC1, and BGL2. You don't have to mug up the sequences and all. Don't memorize it. But I want you to be able in a state to understand the principle of it, the logic or the rationale of the work, and be able to interpret. That is the most important thing. Okay. NOT1 is methylation sensitive. HPA2 is also methylation sensitive. MSP1, methylation independent or insensitive. That means it can cleave either way. BST11 is also methylation sensitive. So you can see that the restrictions are endonucleases that they have chosen. All of them or majority of them have a lot of GC, so that it's easy. They are likely to fall within the CPG islands, right? If you are having CGG, the sequence is simple, CGG, and this one is double CCGG. This one is also CCGG. This one is also G, right? There is a lot of scope for them to be, or they are highly probable that they would fall within the CPG islands, which are imprinted or epigenetically controlled. Okay, we will first work on the H19 gene. Paternally imprinted means the paternal allele is methylated. Okay, it will get a little bit confusing, and don't worry, we are not going to complete this and say that's it. We can always come back and discuss this again. So it would be easy for me to just say this is the result, what they found, and just move on. That's not what we are trying to do. We are trying to understand the rationale and be in a state to interpret. So that's a higher level of your understanding and learning. Here is the setup that they had. They were working with, say, they would have isolated the DNA, and this is the genetic architecture that is present in that location. This is where the probe is, and promoter region, and all the dotted lines, I think, are HPA2. They are indicating HPA2 with dotted lines. BG is for BGL2. Okay. So BGL2 to BGL2. Where is the next BGL? Here. There are also other BGLs here and here, but they're not bothered about it, or they're not mentioning the sizes of the fragment, because the probe would have bound only to this fragment. Only this part would show up in the gel, after development of the membrane with the autoradiography. I'll try to take it again so that we will see what I'll try to take this once again. Let's see. So if I took this DNA, the DNA that is shown here, and I cut it with BGL2, okay, it would give, there is one BGL2 here, there is another here, here, and here. If I would have done electrophoresis, hybridization with the probe, and the probe is indicated here, and develop the autoradiography. What we will find is, if it is only BGL2, then this is the fragment that would be detected by the probe here. So it will show up as one band, a band that is of 5.8 kb. Now, if I treat it with BGL2 plus not 1, right? So BGL2 would cleave here and here. That's the same 5.8 kb fragment. But there is also one not 1 site here. Not 1 will cleave here. So what will we get? We'll get a band from here till here. That is a not 1 BGL2 fragment, which is about 3.4 kb. Okay. So what I want you to also let us look at E is not what we are bothered about. The dotted lines are HPA2. Okay. So if I 1 second, I will see. Not 1 is falling within the, where is not 1? Not 1 is falling within the possibilities of methylation, where it could be highly methylated. BGL2 doesn't have any much of GC. Okay, G and C is there. GC should be together, right? So they are not within the range of where epigenetic regulation would have happened. So I will, before going further, I would want to have a practice exercise so that we will be able to understand it clearly. So if we will have a maternal allele and a paternal allele, right? What bands would you expect if I treated this fragment? We are biparental, right? And okay, I'll just take this. These are the two alleles of this gene. And in this region, which I'm trying to depict here, where not 1 is present, there is methylation. In the other not 1 site, there is no methylation. Here is BGL2 and here is BGL2. The difference between these two is just about methylation of in one of the alleles, and the other one is not methylated. Okay. What would you expect if I treated the fragment with only BGL2? What would we get if I treated with BGL2 and not 1? What bands would we get? Anybody? Can you guess? Should I repeat? So
We will get
BGL2, yes, please.
for unmethylated case, we'll get 2 strands, and for methylated, we'll get 1.
The probe is going to bind only here.
Okay.
Okay. Yes. So which ones will come up in the blot, the end? So this will be cut here, right? Sorry. If it is methylated, not 1 cannot cut. So what will be the size of the fragment then? It will be 5.8 kb from BGL2 to BGL2. In which case, if we had added not 1 also. So, well, I will just go by this. We have taken a sample of the DNA and treated with BGL2. This one, BGL2 and BGL2, will give two fragments of the same size, which would have been 5.8 kb. Nothing different. Both will give the same products. In the second one, BGL2 plus not 1. If I give that, not 1 has a problem, right? It can either cleave or, 1 second, sorry. Not 1 can cleave here, but cannot cleave here. That means what will be the sizes of the fragments? If it is cleaved, this will become about 3.8 kb. From one of the allele, you will get a 3.1 kb fragment, and the other one would be the 5.8 kb. That means not one was able to act on one allele and it was not able to act on another allele because of methylation. Okay, that's how we can figure out if methylation happened or not. Is this understood a little bit? We'll go ahead and try to discuss a little more, and then we'll stop so that you'll get some time to think about it. And in the next class, I will repeat it. Don't worry. So here is the experiment that they have done. Whatever I discussed till now is the preparation for you to understand their work. So every experimental setup is given. HPA, the restriction sites are here. And they have taken samples from four different samples. This is something like MSC Sertoli cells, which is equivalent to that of somatic cells. So are the somatic cells from another cell example. And then here we have spermatogonia. That is something prior to, which is about to undergo gametogenesis or spermatogenesis, but not yet. And here we have mature sperm. Okay. What should we expect is the question. I think we will start here. If it is biparental, like we have discussed here, this is a biparental one, right? And we would get, if we treat with BGL2, we will get one fragment of 5.8 kb. If we treated the DNA with BGL2 and not one, then we'll get two fragments, like here, 3.8 and a whole one, 5.8. So yes, if it is treated with BGL2, then they get one band. That is what we actually is the expected one. If we treat with BGL2 and not one, we are finding 50%. About 50% of the sites got cleaved, and about 50% of the sites did not get cleaved. That could be because one allele is unmethylated or methylated, and one other allele remained unmethylated. There is another one to prove that HPA2 is here. This is the HPA2. All the dotted lines indicate HPA2. So when we treat with BGL2 and HPA2, the HPA2 is highly frequent. That means it is going to degrade all the fragment into very small ones, right? If HPA2 is able to work, I want to just remind you, where is HPA2? HPA2 is also methylation sensitive. Both NOT1 and HPA2 are methylation sensitive. That means if their respective sites are methylated, then it cannot cleave. So what does this mean? That it means that it's not just the NOT1 site that is methylated or unmethylated in this stage. It means, say for example, this band is vanishing, but this band is remaining. Okay, what does that mean? That means HPA2, there are so much frequency here of HPA2 sites, and yet this band remained as such, which means that all this region must have been methylated, at least in one allele. In the other allele, which is which remains as unmethylated, that is where NOT1 has cut, and also HPA2 has degraded the whole of it. There are two alleles, right? One allele has got methylated completely, so much that HPA2 cannot act on it. Other one is remaining unmethylated as it is, and HPA2 will cut it into pieces. so that it doesn't, it becomes very small to be detected here. Okay, that is how we read the results. It is about deduction, and that is how science is usually done. It's like puzzle. Only thing in puzzle is we don't need so much of knowledge about what all the things that are used. In research papers, we should have had some knowledge about what was used by the people, or even if it is your own work, then we should be able to think about those things and plan properly, design the experiments properly, so that you can deduce the answers. Let's see, what would you expect in spermatogonia? In the case of MSC and Sertoli cells, it is biparental, and that means two alleles, one is maternal and one is paternal. Therefore, one could expect a result like this. In spermatogonia, where one would have expected that there is no methylation, right? BGL2, it has nothing to do with methylation, so we don't bother about it. And BGL2 not one alone, you see that most of it is cut. That means the not one site has been cut. That means it is unmethylated in state, and it shows as a 3.4 kb fragment. And you can see that this is very little, but it is still there. Okay, so we are not saying it is 50% or 100%. We are saying mostly in demethylated state. And the same result, it is also confirmed using the HbA2, that majority of this region is unmethylated in spermatogonia. In sperm, where we expect that methylation has been established, and we are talking about U2AF1RS1, which is supposed to be expressed from the paternal allele, it means it should have been unmethylated state, right? H19 is the one, it would have been methylated if it is paternal. Why am I talking it is paternal? Because it's a sperm, and that means it is coming off a male. So BGL2, same thing. In NOT1, we have a problem that there is cleavage. So from spermatogonia, we are going to get the sperm, isn't it? During that process, imprinting must have been finished. In that case, one would have expected that all the DNA would have been, all the site would have been unmethylated, and it should have been cleaved. But the reverse of it is what people are noticing. It is methylated. That's why it is not getting degraded or cut by NOT1. So what we can take from this is, we had a standard model that we discussed a little while ago. The standard model is between somewhere in this stage, all of it is erased or in unmethylated state. And in sperm, at least, you will have that imprint is established, completed. But that's not the truth or complete story of it. It could be more complicated, because the methylation status is not completely done. So if you want to result it, U2F1 is maternally imprinted. That means, for our sake, it was methylated. And in the male, it should have been demethylated. And what observations are found is mostly demethylated in spermatogonia. That is kind of okay. 50% or so is methylated in sperm. And that is something not so well. In what should have happened is, in this case, we have 50% demethylated or 50% methylated. This is the problem that we have. And demethylated in, or sorry, it should be 50% demethylated in the somatic cells. That is fine. Whereas this is the underlined one is the problem. And that means we can only actually, at this point or within the limits of the scope of the paper, we can say that imprint has not yet been established. And there are many other works done by many other labs around the world where there are many of the genes do fall under the standard model, where the primordial germ cells and then erasure and then establishment during the sperm or so gametogenesis. But there are a large number of deviations as well. So that is what is the result they found. We don't have to, for the sake of our learning, we only have to remember that there is a standard model and there is always a probability that there are some variations to it. The important part of the lesson is trying to learn the techniques, trying to be in a state where we can analyze the results, interpretation of the results. So in simple sentence, I think it is from them. The imprinting does not occur during a single cell stage of development, based on what their observations are there in the model organism, in their conditions, in their lab, and in their hands. So we don't really have to take it as a truth, but it's okay to, that is what, there will always be deviations. Like biology is, it's like the science of exceptions, or it's a study, you will have exceptions everywhere. Okay, I will stop here so that you will be able to think upon it and try to visualize. There will be one more of it, but I think one is enough to demonstrate what we wanted to. Technique, the method, and then the result. I will give a problem next time in the exam, so please pay attention to it, and you should be able to deduce. Either you will be asked to draw a gel diagram by giving you a data, or we'll give you a data and ask you to interpret it. Either way, some higher level learning outcomes are expected of you. Try. That is what we can all do. Next class, we will also look at how the chromatin structure could be modified. There are several other ways, methods to study DNA methylation, like one of the sequencing, bisulfite sequencing is there. And there are also histone modifications using chip and so on. And then there is a technique that is, as a part of this syllabus or this lesson, is chromatin structure. I will discuss about it in the next class. Okay, if you have any questions, please ask, so that maybe I'll try to explain it differently. Any questions here? Hello?
Sir, what?
Yes, please.
Okay. We said that HPA2, okay, so yeah, HPA2 cuts the strand into very tiny bits, so it wouldn't come up on the gel electrophoresis. Okay.
Yes.
Uh, if
Yes.
if uh, if they are like cut into small pieces, they are of similar lengths. They'll all get packed in a particular place in the gel, and they
Yes.
will look more dense than not
They will
appear at
form a
all.
band. Is that what you're saying? They would form a band?
Yeah. Yes.
Yes, they might form a band, but it will be in the bottom of the gel because they would be very small. This we are talking in terms of kbs. That could be less than, say, in base pairs, 100 or 200, like that. They would run much faster. Maybe I might have cropped this picture. You might find some degradation products at the end somewhere around here in the bottom of the gel. Okay,
Okay, sir.
we'll go there. Yes. I have evaluated the papers a bit. I think there are 2 more or so. I will try to return the papers on your in the Google Classroom itself. You can